Morning, everyone. We'll just give it 2 more minutes for everyone else to join before we get started. Okay, I think in the interest of time, we will get started, and the recording will be available for those who join late as well. So good morning, everyone, and welcome to today's webinar, Spatial Biology in Focus, High Tissue Resolution Tissue Insights with COMET and RNAscope. So we are really excited to have you join us as we explore how spatial biology is transforming the way researchers understand tissue organization and function. So today's session will focus on two innovative technologies from Bio-Techne, and that is the COMET from Lunaphore and RNAscope from ACD Bio. There may be some time for questions at the end, but if you don't feel comfortable asking your questions in this session, you're always welcome to contact Daniela or myself, and we will be happy to assist you. So it is now my pleasure to introduce the speaker for today. Dr. Daniela De Silva is an application specialist at Whitehead Scientific, and she is really passionate about spatial biology, and so she will guide us through the exciting advances that we have seen in this area of research. So without further ado, I would like to hand over to Daniela. Thank you.
Thank you, Sanisha. Okay, let me quickly share my screen and we can get started. Awesome. Great. So good morning, everyone, and thank you for joining today's webinar. Today, we'll be exploring how spatial biology technologies are enabling researchers to study gene and protein expression directly within intact tissue architecture at single-cell resolution. We'll specifically focus on the two complementary technologies, RNAscope for highly sensitive spatial RNA detection and the COMET for highly multiplexed spatial proteomics. Importantly, we'll also discuss how integrating RNA and protein spatial information can provide a much richer understanding of complex biological systems than either modality alone. So to begin, we'll first establish what spatial biology actually is and why spatial context matters biologically. From there, we'll move into the technical foundations of RNAscope and the COMET individually, before discussing how these technologies work together within integrated spatial multiomic workflows. And finally, we'll look at some real-world disease research applications, as well as practical considerations for implementing spatial biology workflows within the lab. So, traditional molecular biology approaches often require tissue dissociation, meaning that cells are removed from their native microenvironment before the analysis. While these techniques generate extremely valuable molecular information, they also result in the loss of spatial context. And that's where spatial biology addresses this limitation by allowing us to analyze molecular expression directly within intact tissue sections whilst preserving the tissue architecture, the cellular neighborhoods, and the spatial heterogeneity. This allows researchers to understand not only what molecules are present, but also where they are localized and how these cells interact within the tissue environment. So, cellular behavior is highly influenced by spatial context and the surrounding microenvironment. Two cells with very similar transcriptional profiles may behave completely different depending on their neighboring cell populations, their oxygen availability, their stromal interactions, or immune infiltration patterns. So this becomes particularly important in areas such as oncology, immunology, neuroscience, and infectious disease research, where tissue organization itself plays a major role in disease progression and the therapeutic response. Traditional RNA analysis technologies provide powerful transcriptomic information. They generally do not preserve tissue architecture. So, for example, qPCR measures bulk transcript abundance. Bulk RNA sequencing provides average transcriptional profiles, and single-cell RNA sequencing improves cellular resolution but still typically requires tissue dissociation. As a result, important spatial relationships and the cellular interactions may be lost during sample preparation. And that's where RNAscope comes in. This is an advanced RNA in situ hybridization platform developed specifically to overcome many of the sensitivity and specificity limitations associated with your traditional in situ hybridization technologies. The assay enables highly sensitive detection of RNA transcripts directly with intact tissue sections while still preserving histological context and the single cell resolution. One of the major advantages of RNAscope is its versatility. So, in addition to standard mRNA detection, the platform can also be used to investigate viral RNA, splice variants, gene fusions, circular RNA species, microRNAs, and even point mutations. This makes the technology highly valuable across a wide range of research applications, including oncology, neuroscience, infectious disease, and advanced therapeutic development. So, for example, pharmaceutical researchers are increasingly using RNAscope and miRNAscope to study the biodistribution of small interfering RNA and antisense oligonucleotide therapeutics directly within the tissue. Historically, conventional in situ hybridization methods often suffer from poor signal-to-noise ratios, weak amplification efficiency, and limited sensitivity for low abundance transcripts. And this is where RNAscope addresses these challenges through two very key innovations, the proprietary double Z probe design and the branched DNA signal amplification chemistry. Together, these dramatically improve both specificity and sensitivity. The image over here illustrates what RNAscope data can look like in practice, where each punctate dot represents an individual RNA transcript. So in this example, the high-plex assay is simultaneously detecting 12 RNA targets within the same tissue section. So how does it work? The workflow begins with tissue pretreatment and target retrieval to expose RNA targets within the sample. During hybridization, pairs of adjacent probes, known as double Z probes, bind complementary regions of the target RNA sequence. Only when both probes bind correctly can the pre-amplifier molecule attach and initiate the signal amplification cascade. This is a critical feature of the assay because isolated or non-specific probe binding cannot generate a signal, which then significantly reduces the background noise or the staining and improves assay specificity. Following amplification, the transcripts are then visualized using either chromogenic or fluorescent detection methods before imaging and spatial analysis are then performed. So RNA analysis provides important insight into transcriptional regulation, pathway activation, and cellular signaling. However, RNA abundance does not always directly correlate with protein abundance or protein function. So proteins ultimately drive cellular phenotype or biological activity, and this makes spatial proteomics particularly important for understanding the receptor expression, immune cell states, signaling activity, and functional cellular interactions within the tissue itself. And this is where the COMET comes into play. It is a highly multiplexed spatial proteomics platform designed to analyze protein expression within intact tissue architecture at single cell resolution. The system combines oligo-barcoded antibodies, automated fluidics, iterative imaging chemistry, and computational imaging reconstruction to generate the highly detailed spatial proteomic maps from tissue sections. Traditional immunohistochemistry and immunofluorescence workflows are often limited by fluorophore spectral overlap and relatively low multiplexing capacity. So the COMET addresses these limitations through cyclic imaging chemistry and barcode-based detection strategies, enabling significantly higher plex analysis within the same tissue section. The data here is generated using human FFP tonsil tissue following 15 sequential imaging cycles corresponding to approximately 30 protein markers. Importantly, the same tissue section was then subsequently reused for hematoxylin and eosin staining, demonstrating that the workflow remains sufficiently gentle to preserve tissue morphology throughout the entire process. Unlike conventional immunofluorescence approaches, the COMET uses antibodies that are conjugated to unique oligonucleotide barcodes rather than directly conjugated fluorophores. So the reported molecules will hybridize to these barcodes during each imaging cycle, allowing subsets of markers to be visualized sequentially. After imaging, the reporters are then removed and the process is repeated across multiple cycles. The resulting images are then computationally aligned and reconstructed to generate the highly multiplex spatial proteomic datasets from a single tissue section. One of the major strengths of COMET is its ability to characterize complex cellular ecosystems within the tissue itself. So researchers can identify distinct immune populations, activation states, biomarker co-expression patterns, and spatial cellular neighborhoods. This is particularly valuable in applications such as immuno-oncology, translational pathology, and biomarker discovery, where cellular organization strongly influences the disease biology. These technologies provide complementary layers of spatial biological information. So RNAscope focuses primarily on transcriptional biology through spatial RNA detection, whilst the COMET provides functional proteomic and phenotypic insight through multiplex protein analysis. Together, they are able to allow researchers to connect transcriptional activity with functional cellular behavior within the same tissue microenvironment. So integrating transcriptomic and proteomic spatial information provides a much more comprehensive understanding of the entire tissue biology. This is important because multiple biological processes, including RNA stability, translational regulation, protein degradation, and post-translational modification, can influence protein abundance independently of the RNA expression. So this type of integrated analysis allows researchers not only to identify cell populations, but to also understand their functional spatial organization within the tissue. This slide illustrates the overall integrated spatial multi-omic workflow. So following tissue preparation and retrieval, both the RNA and protein detection are performed directly on the COMET platform itself using a fully automated workflow. So for the RNA component, the workflow incorporates the RNAscope high-plex chemistry together with ACD's protease-free pretreatment technology, which allows for highly sensitive RNA detection while still preserving the protein integrity for the downstream multiplex proteomic analysis. Both RNA and protein detection occur through iterative cyclic workflows, after which images are automatically stitched, they're aligned, they're corrected, and then they're exported as standard open microscopy environment TIFF datasets for downstream analysis using either the Horizon or third-party image analysis tools. So spatial biology is becoming increasingly important across many research areas, and this is because biological processes are inherently spatially organized. So in oncology, these technologies are widely used for tumor microenvironment characterization and immune infiltration analysis. In immunology, they support the study of cytokine localization and cellular interaction networks. In neuroscience, they can be used for regional transcript mapping and neuroinflammatory profiling. And in infectious disease research, they enable localization of the pathogens together with the host response characterization directly within the tissue. So this example here highlights how spatial proteomics is beginning to demonstrate translational and clinical relevance. The study shown here involved metastatic castration-resistant prostate cancer patients treated with CXCR2 inhibitor in combination with a hormone therapy. Using highly multiplex spatial proteomics, researchers were able to characterize the tumor microenvironment longitudinally and demonstrate the reduced infiltration of myeloid-derived suppressor cells following the treatment itself. Importantly, the study also showed that spatial proteomics could quantitatively assess the dynamic immune changes within precious clinical samples, further supporting the growing translational value of spatial biology technologies. However, it is important to note that this is currently only for research use. As with any advanced molecular workflow, successful spatial biology experiments depend heavily on experimental design and the sample quality itself. So important considerations always to include within this is the tissue preservation, the probe and antibody validation, panel design, imaging requirements, and downstream computational analysis capacity. So ultimately, high quality spatial data begins with high quality sample preparation as well. The choice between RNAscope, COMET, or an integrated workflow ultimately depends on the biological question being asked. So RNAscope is particularly valuable when studying transcript localization or detecting low abundance RNA species. The COMET is especially powerful for high-plex protein profiling and characterization of complex cellular ecosystems, and increasingly combining both approaches will provide the most comprehensive view of tissue biology itself. To summarize, spatial biology enables molecular analysis while preserving tissue context. RNAscope enables highly sensitive spatial transcript detection. The COMET enables automated high-plex spatial proteomics. And together, these technologies provide integrated multi-omic insight into complex tissue biology. Thank you everyone for joining today's webinar. I hope the session has provided a clearer understanding of how RNAscope and the COMET are helping researchers generate a deeper and more spatially resolved biological insights. I'd be more than happy to take on any questions if there are.
Okay, we do have one question. Let me just unmute the participant.
Right here, I think I'm unmuted. I just wanted to ask, so you mentioned that it's in the cancer example that it's only used for research purposes at the moment. So what is the limitation? What's stopping it from being integrated into a commercial sort of pipeline?
Tanisha, would you mind maybe taking this one?
Sure. So these have not been evaluated as yet in a diagnostic environment, or it isn't marked for IVD purposes. That being said, it doesn't stop it, in the future. But Bio-Techne does primarily focus on research consumables. So, we do, with ACD Bio, we do have some IVD probes, but they are exactly the same as the RUO version. So it is a new technology. It's only come out 2 years ago, so they are still working on a lot of developments. So maybe in coming years, it can be IVD marked, but not enough information is available as yet to do that.
Okay, thank you.
Okay, pleasure. The next person is. Yes, sorry, the next question is from Prof.
Kwan?
Hi, yes.
Sorry.
Thanks for the talk. Melvin Kwan, University of Pretoria. We have a project on DNA viruses, and I looked at the website now. They've got a DNA scope, but I see that it's been discontinued from May 2026. Could we still use DNA? I assume it's just changing the probe, whether you want to detect RNA or DNA.
Yes, so I know you can still do custom probes, but then that would just requires specific sequences, but I can discuss it with Bio-Techne in terms of perhaps designing a probe specifically for the research that you want to do.
Okay, thanks a lot.
We have a question in the chat, Daniela.
Oh, okay, great. Maybe let me stop sharing. I'm going to go into the chat quickly. Fresh tissues when you use either RNAscope, okay, or is FFP tissue the urine type? As far as I know, you're not actually limited to the sample, so I'm pretty sure you are allowed to use fresh tissues. I've seen that this technology can be used for a variety of sample types. It's not limited to just your fixed formalin tissues.
Yeah, I hope that answer helps. Are there any other questions? Okay. I think that is it then. If you don't have the questions right now, or if you want any further information, you're always welcome to contact Daniela or myself. We would be happy to assist. The recording would be available on the Whiteside website, so you can go back to it as well. But yeah, thank you all for joining us and have a great day further.
Thank you,
Bye.
everyone.